Are there any questions? Sorry for being late, but I had to sign in and I had to look up my information because my browser lost the sign-in information. I've been using the browser for a long time and I didn't remember my sign-in information. And then when I got to Zoom, I had to transfer to Clark College and then sign in again. So I had to look up that information too. And then it wanted to send it to my phone to make sure I was actually John Volpe. And I just said, this is just way too much information to get it. And I'm sorry, that's just the way sometimes Zoom and Clark College is. I want to make sure it's me who's signing in, and I don't remember my login information. So when Zoom, the browser didn't fill it in, I had to go look it up, and it took me several minutes to find it. So my apologies for being late. Are there any questions before we get started? Let me state that I've done some grading. I'm not going to look up what it was. So be aware that I have graded at least one of the labs, and it will be, the second grading of it will be due on this Saturday. I think I've graded a large number of the second gradings. I think I did lab 0 through like lab 3 or maybe even lab 4, and they've been graded now for 2 times. I'm not going to look that up, so that's about right. If there's any questions, let me know. We're a little behind schedule because I had so much trouble logging in. But let me state that today is October 31st, and we're supposed to be finishing chapter 5 today. I'm not sure we're going to do that, but hopefully we'll get close. Let me note that you do have an exam, a quiz 3, this Wednesday. Is going to cover labs 5 and 6, and chapter 4, and the end of chapter 3. Any questions about what we're doing? We will have a lab today, and I'm also supposed to send out the results and your unknown number today for your unknown project. And I will do that hopefully either during the lab or shortly after the lab. Any question about any of that? All right. If not, let's begin with chapter 5. Can somebody confirm you're hearing me? Let me check my volume. The volume looks good. Anyone confirm you can hear me?
Yes, we can hear you.
Oh, thank you. Well, let me share my screen. The recording is going. Oh, come on, go down. All right, so I have that we discussed oxidation reduction reactions, and we mentioned in biological systems that the hydrogen atom frequently follows the electron. So in oxidation reduction reactions, also called redox reactions, many animals use catabolism to extract energy from nutrient molecules. And it is true that other organisms besides animals do this, but all animals do it. Green plants can get energy from light and photosynthesis, but they can use catabolism to extract energy from nutrient molecules when there is no sun. And obviously, photosynthetic bacteria can do the same. Highly reduced compounds with many H atoms are degraded, catabolized, to highly oxidized compounds. And in animals, we tend to degrade glucose, C6H12O6, a highly reduced compound with many hydrogens. It's oxidized to CO2 and water. The energy that is released, some of that energy can be trapped in ATP. The highly reduced compounds like glucose contain a large amount of potential energy, and the cell, or our cells, use it to make ATP. And they do that through cellular respiration, which is shown in this slide, glucose plus oxygen, specifically 6 oxygen molecules plus 38 ADP and 38 inorganic phosphates combine to make 6 CO2, carbon dioxide, and 6 water molecules and 38 ATP. This is the overall summary reaction for aerobic respiration. We will talk about each of the steps. Aerobic respiration involves a large number of chemical reactions and some physical reactions. Any question about any of that? In the process of converting the energy in glucose into the energy in ATP and making carbon dioxide and water, some of the energy in glucose is given off as heat. And that's just the second law of thermodynamics. Whenever you change energy from one form to another, some of that energy will be given off as heat. And heat is just another form of energy. Some energy from the redox reactions is used by our cells or by bacteria to make ATP. ATP is generated by phosphorylation. Phosphorylation is just the addition of an inorganic phosphate, PO4 with a -3 charge, to ADP. That's adenosine diphosphate, shown here, ADP plus energy given off by like the burning of glucose and a phosphate make ATP. Let me blow this up. The addition of phosphate makes a high energy bond in ATP, and that's the squiggly line. And you'll note that ATP actually has 2 high energy bonds. If the cell needs energy for some reason, it can break off this phosphate, and that will release a large amount of energy. If the cell still needs more energy, it can actually break off this phosphate, and that will release more energy. And then the cell can use that energy for the cell's energy needs. Any question about any of that? In animal cells, the making of ATP is mainly done by aerobic respiration. In our cells, there's only one other way we can make ATP, and that is by fermentation. And we'll talk about both of these in a little bit. Cells have 3 ways of making ATP. I'm talking about all cells, not just our cells or animal cells. The first way that cells can make ATP is by substrate level phosphorylation, where we have a high energy phosphate bound to some molecule, just represented here by CCC, and then the phosphate, the curved wavy line, meaning a high energy bond. That phosphate can be directly transferred to ADP by substrate level phosphorylation, where the phosphate breaks off of the CCC and then joins the ADP to make ATP. This substrate level phosphorylation actually does happen in both fermentation and glycolysis. We'll talk about glycolysis in just a minute. That's the first step of aerobic respiration. The second way that cells can make ATP is by respiration, also called oxidative phosphorylation. Note that I'm not calling it aerobic respiration because by respiration, we mean both aerobic respiration and anaerobic respiration. It doesn't matter which form of respiration we're talking about. The production of ATP involves the sequential transfer of electrons from an electron donor cell, molecule, such as glucose, to a final electron acceptor, such as oxygen in aerobic respiration. And along the way, that electron is transferred from one molecule to another, giving off some of the energy. And we call this transfer of the electron from one molecule to another. And the electron ultimately starts from glucose through other molecules, which we call the electron transport chain. And energy is released along the way to make ATP by chemoosmosis. And we'll talk a little bit more about that. What I'm going to say is this happens in electron transport chain, the third step of aerobic respiration. Any question about that? As the electron moves from one molecule to another, one molecule will be reduced, the other molecule will be oxidized, and then the electron will move from that molecule to the next. This molecule will become reduced, that molecule will become oxidized. Am I saying that right? Reduction is the gaining of electrons. So that's right. This will be reduced, that would be oxidized. Any question about any of that? The third way that cells can generate ATP is by photophosphorylation. Photophosphorylation only occurs in photosynthesis, where light actually releases an electron from chlorophyll, and that light energy is converted into chemical energy via an electron transport chain. ATP and NADPH are generated. NADPH is similar to NADH. It's just what happens in photosynthetic organisms. They don't generate NADH, they generate NADPH. And then the ATP and the NADPH are often used to make sugar. So that we say that the light from energy will ultimately be trapped into the energy of sugar. And here we see a bunch of organisms which are engaging in photosynthesis. That's why these boys are so muddy. Any question about any of that? That's actually a joke. The boys don't engage in photosynthesis. Don't ask me why they're so muddy, but the plants in the background are engaging in photosynthesis. So we have metabolic pathways for making energy. And for animal cells, the energy is released from molecules in controlled ways and stored in a series of reactions, meaning that the energy is not released in a single burst. We could release the energy in glucose in a single burst. And that is if you have a cup of glucose and then you bring a blowtorch to it, you can create the, burn the glucose and it'll start burning itself. And so much energy will be released from glucose that you'd have to stand several feet away. And then you'd have to blink your eyes or shut your eyes because the light would be so intense and the heat would be so intense you need to get far away. Obviously, animal cells and other cells do not release the energy in glucose in a single step because a blowtorch is something like 1,000 degrees. And I don't remember if that's 1,000 degrees Fahrenheit or 1,000 degrees Celsius. It doesn't matter. It's very hot, and our cells could not survive that temperature. Instead, electrons from the donor molecule, such as glucose, are passed from one compound to another through a series of redox reactions, releasing the energy in glucose in small steps. And as you know, our cells maintain about 37 degrees Celsius or 98.6 degrees Fahrenheit. Even when we're engaging in aerobic respiration, the burning of glucose. So that's happening much lower than 1,000 degrees Fahrenheit or Celsius, whatever that is, 1,000 degrees. Well, let's just say it that way. Most organisms can use carbohydrate catabolism to obtain their cellular energy needs. And many organisms, like actually all animals and many bacteria, can use carbohydrate catabolism as their primary source for obtaining cellular energy. Even green plants can use carbohydrate catabolism, the burning of glucose, to generate their cellular energy needs. And if you don't know, green plants actually do that at nighttime. Or if you're a green plant in Point Barrow, Alaska, when the winters are 3 months long, the green plants are using carbohydrate catabolism for their energy needs in the wintertime. But even in the nighttime, assuming you don't have a night light like your plants outdoors or something, if they need energy, they will engage in carbohydrate catabolism to get their energy needs. Obviously, when the light is around, green plants and photosynthetic bacteria can get their energy needs from the light. The most common energy source for carbohydrate catabolism is glucose. And generally, if you're a couch potato like me, we will convert whatever other food we consume, we will convert that into glucose and then send glucose down the aerobic respiration in the normal way. You can directly burn like lipids and proteins, like marathon runners can directly burn lipids in aerobic respiration, but they don't do that until the marathon runner has actually ran for 45 minutes. Initially, the marathon runner is only burning carbohydrates to generate their energy needs. And they will slowly, although they're not being a couch potato, but they'll slowly convert lipids into glucose. The trouble is they'll soon run out of the glucose, and then they have the lipids that they can burn, and the marathon runner will directly burn the lipids in aerobic respiration, but they don't do that until 45 minutes into their run. Any question about that? The point is lipids and proteins can be utilized in aerobic respiration, but most of us couch potatoes, we will burn the lipids and proteins by changing them into glucose and then sending glucose through aerobic respiration. And actually, all of the carbohydrates that I'm aware of, if they're not glucose, they'll be converted into glucose. And then glucose will enter the aerobic respiration. To produce energy from glucose, microorganisms use two general processes to make ATP. First, there's a respiration, and you'll note I'm saying not aerobic respiration because it's both aerobic respiration and anaerobic respiration. The second way that cells can generate ATP from glucose is by fermentation. And we'll talk about all of these ways that cells can generate energy from glucose. But the main way we're going to talk about and spend most of the time on is aerobic respiration. Any question about any of that? If there's no questions, let's go ahead and watch a video on aerobic respiration.
Once you see what's in these two
after
heavy-duty
the
totes,
commercial,
you'll feel safer when the next disaster rolls around, because you'll know.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions about any of that? All right, not hearing anything, so let's continue. Where am I? There we go. So let's talk a little bit more about aerobic respiration, and we're going to compare it with fermentation. This slide is showing you the overview of aerobic respiration. It has 3 principal stages: glycolysis, the first step, the Krebs cycle, the second step, and then the electron transport chain. Essentially, electrons from glucose, an energy-rich molecule, are used to make ATP, and there's a high yield of ATP in aerobic respiration, and then the glucose is converted into CO2 and water. Both of these are energy-poor molecules. Compare that to fermentation, which also uses glucose to make ATP. Fermentation is only 2 steps. Glycolysis does occur, and then there's a fermentation step, which can be 1 or 2 chemical reactions. There is no Krebs cycle, no electron transport chain. Instead, pyruvate or pyruvic acid is converted into the end product of fermentation. There's a much lower ATP yield for fermentation compared to aerobic respiration. Any question about any of that? All right, this is an overview. I guess I'll leave this as a big slide. Here in the pink, you can say that's a eukaryotic cell. I know that's the case because there's the mitochondria, so this is a eukaryotic cell. It's showing you the overview of aerobic respiration in a eukaryotic cell. Glycolysis occurs in the cytoplasm. Glycolysis converts glucose into pyruvate. Some NADH is made. The electrons from glucose are being carried by NADH. And the NADH comes to the third step, which I'm going to call the electron transport chain. Third step of aerobic respiration. We'll get there. The pyruvate then enters the mitochondria of a eukaryotic cell and enters the preparatory step, and then the Krebs cycle. Here it's called the citric acid cycle. There is some making of ATP. I should mention there's some making of ATP in glycolysis too. And then the carbon in glucose is converted into CO2 at the preparatory step right here and the Krebs cycle right here. Some ATP is made, but a lot more NADH and some FADH2 is made in the Krebs cycle. The NADH and the FADH2, as well as the NADH from glycolysis, carry the electrons that originally came from glucose to the electron transport chain, the third step of aerobic respiration. And in the third step of aerobic respiration, ATP production is goes into high gear. We get a lot of ATP being produced in the electron transport chain. Now, as I stated, pyruvate moves into the mitochondria of a eukaryotic cell. And then the Krebs cycle and the electron transport chain occur either inside the mitochondria, or for the electron transport chain, it occurs across the inner wall of the mitochondria. That is the overview of aerobic respiration in a eukaryotic cell. In a prokaryotic cell, everything happens in the cytoplasm because there is no mitochondria. And then the electron transport chain happens across the cell membrane. And we'll talk a little bit more about that. Any questions about this overview? All right, let's end the show here. Glycolysis does have another name. I don't use it. I'm not even sure how you pronounce it, but I'm going to guess it's something like Embden-Meyerhof pathway, after two doctors who discovered the steps of glycolysis, and they're German, and I might not be pronouncing their names correctly. In glycolysis, we have the oxidation of glucose to pyruvic acid. Glycolysis occurs in most living cells. It can occur in the presence or the absence of oxygen. Some ATP and some NADH are made, but not a lot. Any question about that? All right, let's talk about glycolysis in a little more detail. Let me blow this up. Here we're seeing the chemical reactions involved in glycolysis, starting from glucose up here, there it is there, converting into this molecule, that molecule, that molecule. You note some ATP is used up. And then we have this molecule converted into this molecule and that molecule. This molecule will be converted into that one. And then this molecule converted into that one, that one, that one, that one, and that one. So it's like 9 or 10 steps to convert glucose into pyruvic acid, which is similar to pyruvate. You need a chemist to tell the difference. And the cell uses pyruvic acid the same as it uses pyruvic acid. Pyruvic acid, the same as it used pyruvate. So we'll just use the two terms synonymously. We get 2 ATP here and 2 ATP there, but we put in 2 ATP or use up 2 ATP in glycolysis. So we say that we get a net gain of 2 ATP out of glycolysis. when I was an undergraduate, I had to memorize each of the intermediate reactions, reaction product, I guess you'd call it, as well as all of the enzymes for making each step of glycolysis. And since I was required to memorize that, I think it's fair that I require you guys to memorize that. What do you guys say about that?
No, thank you.
You only need to know that glucose starts out glycolysis. So you start with glucose, and then you end up with pyruvate. Where the heck is that? Or pyruvic acid. You do get a net gain of 2 ATP, 2 NADH molecules out of glycolysis, and then 2 pyruvates, because pyruvate or pyruvic acid are 3 carbon molecules, and glucose is a 6 carbon molecule. Any question about any of that? So let me remind you, respiration is the oxidation of molecules to release electrons, which will be used in an electron transport chain to generate ATP. There are 2 forms of respiration: aerobic respiration, where the electrons will travel to oxygen. So the final electron acceptor is oxygen. But there is anaerobic respiration, where the final electron acceptor is not oxygen. It's some other molecule other than oxygen. ATP is generated in both steps by oxidative phosphorylation, mainly in the electron transport chain. All right, so the pyruvic acid, also called pyruvate, that is generated in glycolysis cannot be directly used by the Krebs cycle. So it goes through the preparatory step, which is one chemical reaction to convert pyruvic acid to acetyl coenzyme A. So coenzyme A attaches to form acetyl coenzyme A, and now the acetyl coenzyme A can directly enter the Krebs cycle, the next step of aerobic respiration. I should mention that the preparatory step does generate 1 NADH right here, and pyruvic acid is a 3 carbon molecule. Acetyl coenzyme A is a 2 carbon molecule. So we get the first carbon of glucose that is made into CO2. Now I should mention that for each molecule of glucose, you will get the preparatory step running twice, because that each molecule of glucose will generate 2 pyruvic acids, and both of them will go through the preparatory step. Any question about any of that? All right, the next step of aerobic respiration is called the Krebs cycle. The Krebs cycle has a couple of different names. Sorry about that. It's called the Krebs cycle after Dr. Krebs, who discovered it. It is also called the tricarboxylic acid cycle, or TCA cycle, because it has a lot of tricarboxylic acid. That's 3 carbon units. It's also called the citric acid cycle, which is conveniently abbreviated TCA for the citric acid cycle. And it's called the citric acid cycle because citric acid is the first molecule made in the Krebs cycle. I and your textbook tend to call it the Krebs cycle. Any question about any of that? All right, this reaction is showing you, wrong one here, well, showing you both the preparatory step, which I've already discussed, and the Krebs cycle. Pyruvic acid in the preparatory step is converted into acetyl coenzyme A. Acetyl coenzyme A then enters the Krebs cycle. It binds with oxaloacetic acid to make citric acid. Why the Krebs cycle is sometimes called the citric acid cycle, because citric acid is the first product made in the Krebs cycle. Citric acid is then made into this molecule. This molecule is then made into that molecule. That molecule is then made into that molecule. And in the process, we make some ATP. This molecule is made into that molecule. That molecule is made into that molecule. This molecule is then made into oxaloacetic acid, which is where we started in the Krebs cycle. Why it's called the cycle? Because as long as acetyl coenzyme A enters this cycle will continue for as long as you are alive. Any question about any of that? So in the preparatory step and the Krebs cycle, we generate one ATP that happens down here. Come on, mouse. Sorry, my mouse has died. Oh, there it goes. Right there. We generate 4 NADH, one I've already mentioned in the preparatory step here, one in the Krebs cycle here, another in the Krebs cycle there, and the fourth in the Krebs cycle here. We do generate one FADH2 right here, and then we generate 3 CO2s, one here in the preparatory step I've already talked about, one here in the Krebs cycle, and the third here in the Krebs cycle, meaning all of the carbons in pyruvic acid are burnt into CO2 because glucose generates 2 pyruvic acids. For each molecule of glucose, we will run the preparatory step and the Krebs cycle 2 times, so that each molecule of glucose having 6 carbons will be burnt into 6 CO2. So 2 times 3, 1, 2, 3 will be 6 carbon dioxide. So each of the carbons in glucose will be burnt into CO2. And we will breathe that CO2 out, which is why we exhale. And that's to exhale the CO2 that is generated from aerobic respiration. Any question about that? And I guess the other thing I should mention is none of that CO2 comes from oxygen. All of that CO2 originally came from glucose. Any question about any of that? So you should memorize what's in the red box. In the preparatory step and the Krebs cycle, we generate 1 ATP, 4 NADH, 1 FADH2, and 3 CO2. And then remember, the preparatory step and the Krebs cycle runs twice for each molecule of glucose. So if I ask you the question, how many ATP are generated in the preparatory step and the Krebs cycle per molecule of glucose, you will take these numbers and multiply by 2. So it would be 2 ATP that are generated per molecule of glucose. We will talk about the NADH and the FADH2 in just a little bit. They're destined for the electron transport chain. And let me reiterate, this is called the Krebs cycle because it's a cycle. We end up at the same molecule we began the cycle with. So it goes in a circle. Any question about the preparatory step in the Krebs cycle? All right, so we've talked about glycolysis, the first step of aerobic respiration, and now we talked about the preparatory step and this Krebs cycle, the second step in aerobic respiration. Both of these steps generate some NADH, and the Krebs cycle generates some FADH2 as well. Those carry the electrons that ultimately came from glucose, and the electrons will be taken to the electron transport chain to generate more ATP. Any question about any of that? All right, aerobic respiration. The third step of it is the electron transport chain. It does have some other names, which I don't use. One of the other names doesn't tend to confuse students, and it can also be called electron transport phosphorylation. That's similar to electron transport chain. I will call it the electron transport chain, and your textbook calls it that also. But it can be called chemiosmosis and oxidative phosphorylation. Like I said, I don't use those terms because they're so different from the electron transport chain that it's hard to remember all the names for the electron transport chain. Any question about any of that? Excuse me here. Hopefully you're not seeing that. The electron transport chain is a series of carrier molecules that are in a membrane, and they are in turn reduced and oxidized as electrons that come from glucose are passed from one molecule in the chain down the chain to the next molecule. Energy is released as the electrons are transferred from the high energy glucose molecule to the lower energy compounds such as CO2 and water. Energy is released by chemiosmosis to produce ATP. And let's watch a little video on the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about that video? Can't get it. All right, let's skip it. All right, aerobic respiration. The third step is the electron transport chain. This occurs along the inner mitochondrial membrane of eukaryotic cells. And in prokaryotes, it occurs along the cell membrane. So that happens on the inside of that membrane and then on the other side of the membrane. And like I said, with eukaryotes, that membrane is the inner mitochondrial membrane. For prokaryotes, that membrane is the cell membrane. Any questions about any of that? All right. So what happens is NADH and FADH2 brings the electrons. Ultimately, the electrons came from glucose. And the electrons are given to the proteins in a membrane. And that's why it's called the electron transport protein. The electrons are given to a protein, which is in a membrane, and then the electrons transport from one protein to the next, to the next, to the next, to the next, until they leave the electron transport chain. And with aerobic respiration, those electrons flow to oxygen. Oxygen takes the electrons and some hydrogen ions to make water. So that's why it's called the electron transport chain. It's a chain of proteins in a membrane. For eukaryotes, that's the inner mitochondrial membrane. For prokaryotes, that's the cell membrane. And then the electrons transport from one protein to another in the membrane. As the electrons move through some of the proteins, this protein here, come on, mouse. This protein here, this protein here, and this protein here, the electron gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. You'll note that NADH is a little more efficient, and that is it gives the electron to the first protein in the membrane, whereas FADH2 is not as efficient. It gives it to the second protein in the membrane, and so it's missing the first protein, and the first protein is a protein that can pump hydrogen ions from this side of the membrane to that side of the membrane. When the proteins pump hydrogen ions from this side of the membrane to that side of the membrane, that'll make fewer hydrogen ions on this side of the membrane and more of them on that side of the membrane. That creates potential energy difference. There will be a twofold charge or two charges making potential energy, and that is first, this side of the membrane will be more positively charged than this side of the membrane. And that's because hydrogen ions have a positive charge. The second thing is there will be more hydrogens on this side of the membrane than on that side of the membrane. So these hydrogen ions will want to flow from areas of high concentration, which is this side of the membrane, to areas of low concentration on this side of the membrane. That's just the laws of thermodynamics. However, the hydrogen ions cannot cross this membrane except at this channel protein. This protein is an ATP synthase protein. And when the hydrogen ions move through this protein, it gives the protein the energy it needs to make ATP by converting ADP, an inorganic phosphate, into ATP. And so this potential energy difference is used to make ATP. Any question about any of that? All right. This is why we have to breathe in oxygen. When we breathe in oxygen, all the oxygen we breathe in and utilize is converted into water. None of that oxygen we breathe in is converted into CO2. All of that oxygen is converted into water. For humans, a lot of this water will be breathed out. However, for desert animals, a lot of that water will be used to supply the desert animals' water needs. I think like desert mice, for example, 65% of the water they need comes from aerobic respiration. And we can use that water too. So not all of it is wasted. A lot of water that we generate in aerobic respiration is used for our water needs. But we're not as good at conserving water as a desert animal. And so we don't get 65% of our water needs from aerobic respiration. Any question about any of that? All right. So that's why the cell pumps hydrogen from this side of the membrane to that side of the membrane. And how it does that is from the energies that come, the electrons that come from glucose. They're ultimately used to pump hydrogen ions from this side of the membrane to that side of the membrane. And then the hydrogen ions are used to make ATP. The electron then flows to oxygen. So oxygen is the final electron acceptor, and it combines the electrons with hydrogen ions to make water. Any question about the electron transport chain? All right, let's talk about the number of ATP that are generated in the electron transport chain. In prokaryotes, they're a little more efficient. They generate 34 ATP per molecule of glucose by the electron transport chain of aerobic respiration. And that's important too. I need to say of aerobic respiration, because at the moment, we're only talking about aerobic respiration. Eukaryotes are not quite as efficient. Most eukaryote cells can only generate 32 molecules of ATP in the electron transport chain of aerobic respiration. However, we do have 3 cells in our body which are more efficient at generating ATP, and they can generate 34 molecules of ATP per molecule of glucose. The cells which generate the 34 molecules of ATP in the electron transport chain are the cells of your heart, the liver, and the kidney. Does anyone have a guess why your heart is more efficient at generating ATP than, let's say, your skin cells, the cells of your nose, the cells of your bone, the cells of your brain? Anyone have a guess? Well, as is obvious, your brain cells are not working right now because nobody's guessing. So your brain cells and your skin cells they're not always working, so they don't always need ATP. The cells of your heart, does it ever rest for any length of time? More than, I don't know, a minute or two?
Hopefully not.
Yes, hopefully not. Not until you die does your heart stop working. So your heart, your kidneys, and your liver, are always working. And so those cells are more efficient at generating ATP than all the other cells in your body. Any question about that? You already talked about the final electron acceptor in aerobic respiration is oxygen. And let's talk about this in a little more detail. There's the overview of aerobic respiration. This is showing you how the electrons are carried. They ultimately come from glucose, and they're carried to the electron transport chain by NADH or FADH2. And then the energy in those electrons is given off step by step as the electron is passed from one protein in the membrane to another, until ultimately the electron leaves the last protein, and in aerobic respiration, that electron will flow to oxygen. And this is showing you where the steps of the electron transport chain occur in both a prokaryote cell, let me blow that up, and a eukaryote cell. In a eukaryotic cell, these proteins in this membrane, that membrane is the inner membrane of the mitochondria. And so the hydrogen ions are pumped in between the inner membrane and the outer membrane, so that this really helps protect the cell, because in eukaryotes, those hydrogen ions aren't just free floating. They're between the inner membrane and the outer membrane of the cell. Any question about any of that? In prokaryotes, this membrane shown here, that's the cell membrane. So these electrons are pumped from inside the cell or in the cytoplasm to across the membrane to where? Where are they pumped in in most bacteria? Where are these hydrogen ions pumped? Outside the cell membrane, but what's right here? Come on, it's right there, people.
The cytoplasm?
No, they're pumped from the cytoplasm across the cell membrane outside the cell. So these hydrogen ions are actually pumped into the cell wall. You got that? In a prokaryote. And that's safe for the cell because this is actually outside the cell in the cell wall. The cell wall doesn't do anything to the hydrogen ions because hydrogen ions can dissolve in water, and water can move the hydrogen ions right through the cell wall. All right, I already talked about that, although I haven't called that the pumping the hydrogen ions. It's called the proton motive force. I don't talk any further about that, and I won't quiz you on that either. And there's another term for the electron transport chain. Chemiosmosis refers specifically to the making of ATP. And oxidative phosphorylation is another name for the electron transport chain. I don't use this term because it's so different from the electron transport chain. It'll just confuse people. I'm just introducing it in case you have a different author who uses it. All right, so let's do the counting of ATP and aerobic respiration for glucose engaging in aerobic respiration. The prokaryotes generate 38 ATP molecules. That's 2 in glycolysis, 2 in the preparatory step in the Krebs cycle, and 34 in the electron transport chain. For eukaryotes, they also generate 2 in glycolysis, 2 in the preparatory step and the Krebs cycle, and then 32 to 34 in the electron transport chain, depending on what cell we're talking about. So that eukaryotes generate a total of 36 to 38 molecules of ATP per molecule of glucose, and prokaryotes generate 38 molecules of ATP per molecule of glucose. And once again, here's the overall summary equation for aerobic respiration, where glucose, C6H12O6, plus 6 oxygen molecules, plus 38 ADP, and 38 inorganic phosphate, generate 6 carbon dioxide, 6 water molecules, and then 38 ATP molecules. Any question about any of that? All right, aerobic respiration. I've already talked about this, so I'm not going to go further. Let's briefly talk about anaerobic respiration. Anaerobic respiration is when the final electron acceptor in the electron transport chain is a molecule other than oxygen. It can be, or it is an inorganic molecule. It can be nitrate or sulfate. There are different forms of anaerobic respiration, and they each have a different final electron acceptor. Glycolysis and the Krebs cycle still occur in anaerobic respiration. The protein chain of electron transport chain is shorter in anaerobic respiration than in aerobic respiration, so the yield of ATP is less than the yield of ATP in aerobic respiration. The yield of ATP for anaerobic respiration varies between 2 molecules of ATP per molecule of glucose up to 36 molecules of ATP per molecule of glucose. So for prokaryotes, anaerobic respiration is not as efficient, and therefore anaerobes tend to grow more slowly than aerobes. And that's just because the anaerobe cannot generate as much ATP per molecule of glucose as an aerobe can. Any question about any of that? As I mentioned, there are different forms of anaerobic respiration. The best forms of anaerobic respiration generate 36 molecules of ATP. The very worst form of anaerobic respiration, depending how you define anaerobic respiration, will either be 2 molecules of ATP or 4 molecules of ATP. Okay? Any question about any of that? And I don't want to get into what's the definition of anaerobic respiration, so we're not going to get into why some authors say it's 4 and some authors say it's 2. We'll just say it's at least 2 up to 36. All right, let's talk a little bit about fermentation. That is another way that cells can generate ATP from glucose. Fermentation always involves glycolysis, and then it has 1 or 2 fermentation steps after glycolysis. Fermentation does not yield as much ATP as aerobic respiration. It does release energy from oxidation of an organic molecule like glucose via glycolysis. Glycolysis proceeds previously as discussed, producing 2 pyruvic acid molecules, a net gain of 2 ATP, NADH, and we're not going to talk about the hydrogen ion, from the oxidation of glucose. Other carbohydrates can be used, as well as amino acids and lipids, and then nucleotides can engage in fermentation. Let me see, when's the time when we use something other than glucose? Well, when you put barley in is that beer or wine they make out of barley? I don't remember. I think it's beer that uses barley. When you put the barley in, it's mostly the starch of the barley that will be fermented, and the fermentation will generate ATP. And so in that case, we're using mostly starch of glycolysis in fermentation. So fermentation involves glycolysis, where we generate 2 ATP molecules, or a net gain of 2 ATP. All the ATP is generated in fermentation in glycolysis. We also generate 2 NADH. We do not have the Krebs cycle or the electron transport chain. Instead, the electrons that ultimately come from glucose are taken by an organic molecule, such as pyruvic acid, to make the end product of fermentation. In fermentation, we take the NADH made by glycolysis to generate NAD+. We're about to run out of time. I normally like to ask students, why do you think our muscle cells, which can engage in fermentation, why don't they retain that NADH instead of using it in fermentation? Because it just converted into NAD+, we don't make any ATP out of this NADH in fermentation. It's just used here. And it would be better for the cells to just keep that NADH around, because our muscle cells will eventually be given oxygen and, more glucose from the blood. The heart and the lungs are working like crazy to bring oxygen to the muscle cells, but it can't be brought fast enough, no matter how quickly the heart and the lungs work. Your muscles will always use oxygen faster than it can be brought to the muscle cell. And so the muscle cell will fall back on fermentation to make ATP. But my question is, why doesn't the muscle cell just keep that NADH and wait for oxygen to come back to the muscle cell? Because it can make more ATP from aerobic respiration than it can from fermentation. And the reason is the cell has to have NAD+ for glycolysis to occur. And so the cell uses this NADH in fermentation to regenerate NAD+, so that the cell will have that NAD+ to engage in glycolysis to make ATP. If there's no NAD+ around, the muscle cell cannot make ATP. If the muscle cell has no ATP, the muscle cannot work. And if you don't know, if there's the muscle cells will not work. Like an Olympic athlete, every Olympics, we usually have one. They use up all of the glucose in their muscle cells, so their muscle cells cannot generate any ATP by even fermentation, and the Olympic athlete just collapses. In the wintertime, the coach runs out and throws a blanket over them. In the summertime, I assume they run out and squirt them down with water just to help keep them a little cooler. But then the Olympic athlete will get up and then walk across the finish line because the heart and the lungs are working like mad to bring oxygen to the muscle cells. And once oxygen is brought to the muscle cells, aerobic respiration can resume. The Olympic athlete will make ATP, and then their muscles will start working again. Any question about any of that? All right. 2 fermentation reactions I do want you to know. There are a couple of fermentation reactions, but 2 you should know is lactic acid fermentation. That's where glycolysis produces 2 ATP, and then pyruvic acid is converted in 1 fermentation step into lactic acid. This is actually a common fermentation step that many bacteria have. Our muscle cells have it, so it builds up lactic acid. And there's a lot of foods that use the lactic acid to prevent the food from spoiling, such as sauerkraut, yogurt, what do you call that? Kimchi, or, well, sauerkraut. I got it right there. Never mind. Sourdough bread can use lactic acid fermentation, although sourdough bread also uses alcoholic fermentation. So a lot of foods, like most cheeses, use lactic acid fermentation. Yogurt uses it, sauerkraut uses it, sausage uses it, even your favorite chocolate. Chocolate uses lactic acid fermentation. Another fermentation reaction I want you to know is alcoholic fermentation, where glycolysis produces 2 ATP, and then in 2 chemical reactions, pyruvic acid is converted ultimately into ethanol. Ethanol is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So CO2 is given off in the alcoholic fermentation. And in a bottle of beer, that CO2 gives the beer its fuzz. In wine, the wine is made in an open barrel or open casket, and the CO2 just is shed to the air. However, if you were to capture that CO2, like when you were to bottle the wine and then ferment it in a closed bottle of wine, we don't call it wine, we call that champagne. And that champagne, the CO2 generates the bubble in the champagne. There are different fermentation reactions. Let me blow this up. Lactic acid fermentation and ethanol fermentation happen by many bacteria, or ethanol by yeast. Swiss cheese actually uses propionic acid fermentation, and that generates CO2. The CO2 creates the hole in the Swiss cheese. Why Swiss cheese has a hole or has holes in it. Some of these other fermentation reactions are used in industry to make like butanol or butyric acid, lactic acid or succinic acid, lactic acid, formic acid, Enterobacter. I think that one's used to make, what do you call that? Vinegar. Anyways, don't memorize this slide. I just wanted to tell you that there are a handful, about 6 different fermentation reactions. The most common is the lactic acid fermentation, which, like I said, many different foods use that one. The next most common probably is the alcoholic fermentation, and bread uses that. Leavened bread uses that. Beer and wine use that. This reaction is showing you some of the different types of fermentation reactions, the industrial or commercial use. You don't need to know this slide. The starting material, and then the microorganism engaging in the fermentation, like lactic acid, Lactobacillus coccus. These are bacteria, and Lactobacillus delbrueckii. These are bacteria we use to make yogurt, for example. All right, any questions about fermentation? Let me summarize fermentation, then I'll end the lecture here, and that is in aerobic respiration compared to anaerobic respiration and fermentation, the growth conditions of aerobic respiration is aerobic, meaning you have to have oxygen around for aerobic respiration. Anaerobic respiration is anaerobic respiration, and that's because it happens without the presence of oxygen. Fermentation can happen either in aerobic conditions or anaerobic conditions. Oxygen is not required, but it doesn't hinder fermentation. I should mention that ATP production or aerobic respiration is 36 to 38 in eukaryotes, 38 molecules of ATP in prokaryotes. And anaerobic respiration is between 2 and 36 molecules of ATP. In fermentation, it's only 2 molecules of ATP. Any question about any of that? All right, I'm going to stop it here, and we will continue in the lab at 6:30. Are there any questions before I log out?
Nope.
All right.
Thank you.
All right.